Welcome to ESM 101 Lab. My name is Alex Staunch and I'll be the graduate teaching assistant for this course. So basically I'll be administering, narrating the lectures. That could be a good or bad thing. As well as I will be monitoring discussions, grading any material, and also fielding any questions or concerns that you have for the lab section specifically. So you can find my email on the D2L module. It's right in the syllabus, as well as you can message me directly on D2L. So I look forward to working with you throughout the quarter, and hopefully you get a lot out of this lab as well as the ESM lecture. Thanks. So let's jump right into things here. With the first lecture for the lab, we'll be discussing ecosystems and how biodiversity is composed and made up within these ecosystems. So the learning objectives here, which you can find in the document on D2L for week 1. Basically, out of this PowerPoint, I would like you to be able, by the end, to define an ecosystem and also explain the benefits as well as why ecosystems are necessary for human survival. I would like you to be able to define biodiversity as well as the 3 different scales of biodiversity, which we'll be getting into. And then also explain the technical difference between species richness as well as evenness. And when we're going through these lab PowerPoints and lectures, I want you to always be thinking about how you can incorporate what you're learning in these lectures to your field sites or the overall study that you'll be carrying out throughout the length of this course. So, ecosystems as a definition. So ecosystems can be seen as a biological community of interacting organisms and the physical environment. So you can see this as 3 different components. You have your biotic, or all your living things. Everything from the big bears in the woods, the birds in the skies, the huge trees, the insects, the fungi, and the bacteria. Anything alive is incorporated in the biotic community. And then the physical environment can be seen as anything not alive or abiotic. So that incorporates soil, water, energy influx from the sun, air quality, anything and everything that would affect living organisms. And I just love how now the abiotic elements are not only earth, air, fire, and water, but also pollution. So that also affects biological communities and ecosystems as a level of pollution. So here we have the word of interacting. So an ecosystem has the 2 components of biotic and abiotic, but it's how they incorporate and interact with one another that truly makes an ecosystem. So examples of this could be a wetland ecosystem, a desert ecosystem, a montane ecosystem, a freshwater lake ecosystem. So again, we're looking at the environment as well as the organisms within that environment and how they interact with one another. So what people don't realize is how important ecosystems are to mankind. Not only in the fact that it's nice to have parks to go visit, it's nice to feel good that you're conserving nature, but their economic value is huge. So ecosystem services that are provided to humankind can include such things as water supply. So we utilize the ground to filter our water and make it drinkable. We don't need to treat all the water that hits the Earth's surface because ecosystems and Mother Nature supplies this service for us, as well as soil formation. If microbes didn't break things down, if physical erosion didn't occur, we'd have to make our own soil. But no, Mother Nature does do that. So all these services have been seen to add up to $33 trillion a year, the economic value to humankind. And one example of this is pollination. One big issue these days is the disappearance of a honeybee, or even the extinction of other pollinators such as birds and other insects. Think about how much it would cost to have to hand pollinate all of our crops, as well as natural vegetation in forests or other ecosystems. So it's important to look at the fact that ecosystems do have an economic value, and that is just one of the many reasons why we have to start conserving them. So biodiversity. Biodiversity can be seen as the complete range of species and biological communities, as well as the genetic variation within species and all ecosystem processes. So we're going to be talking about biodiversity here in three contexts. So we're going to be looking at the total number of species, so all the species on Earth, as well as you can say how diverse is this park? How diverse is the state of Oregon or Massachusetts by merely counting the number of species there. And then we also look at how diverse is one species genetically. How many different genes will code for the same feature or character in a species. And one example of this is cheetahs. Cheetahs were basically bottlenecked, or the whole species was dwindled down to one pregnant female about 10,000 years ago. And that one pregnant female yielded all the genes to all of the cheetahs from here on out. So therefore you have almost all cheetahs look pretty much the same. There's little physical variation because there's little genetic variation. And also there's large problems with breeding, which comes from that. But then when we look at ecosystem processes, so there's a diversity in that in itself. So you look at all the different environments out of there. The state of Oregon can be seen as being quite diverse in this way, in the fact that we have an ocean environment, an ocean ecosystem, as well as mountains, rainforests, deserts, plains, grasslands. Basically, there are a huge amount of microclimates or small ecosystems in the same area, which yield a large amount of species and genetic diversity. So as we touched upon, species diversity can be defined as the variety and abundance of the organisms which inhabit an area. And you'll definitely be utilizing this in your field sites, where you'll just be basically identifying and counting the number of plant species, or possibly insects if you choose to carry out a site such as that. And then ecosystem diversity. So it encompasses a variety of habitats that occur within a region. So as I talked before, you can have an ocean environment, or here are the painted hills, kind of the grasslands or desert area in Oregon. And this looks at all the different types of forests within Oregon. And every different type of forest yields different organisms. They have totally different communities, therefore they are totally different ecosystems. And then genetic diversity can be seen, again, as the combination of different genes found within a population of a single species, and the pattern of variation found within different populations of the same species. So you have two different cases here. Genetic bottlenecking, kind of the same issue that I talked about with the cheetah. Otters, they were hunted almost to extinction, so therefore you had just a small population of otters that then basically yielded the much wider and larger population of otters that we see today that came about from conservation. But you went from this huge population of otters with a large amount of genetic diversity and dwindled it down through hunting to almost nothing. So the number of genes that exist in the population are basically a fraction of what they used to be. And then here with orca whales, you're seeing actually diversification or speciation, the fact that they're finding groups of whales that are feeding on different, basically on different sources. You have some that go for shells and basically forage on the ocean bottom, and they stay closer to shore, while you have whales way further out in the ocean that feed on fish and some seals and other larger organisms, and therefore their behavior is different. They are not breeding with one another, and they're actually finding that their jaws are changing, and there's actually physical differences forming within these, you could say, two separate populations of orca whales. So what drives that behavior and also that physical change is genes. So the gene pool is widening within the species of orca whales as a whole. And so the two components of biodiversity would be seen as richness and evenness. And richness is simply the count of species within an area. And if we go and we refer to this simple table right here, I just want to let you know that a plot, whenever I refer to a plot, it is simply an area, anything. It could be seen as an acre, it can be seen as 10 square feet, it can be seen as a park. It's just one area in which we are collecting data. In this case, we are counting plant species. So richness can be seen as, in this case, in plot number 1 and plot number 2, the species richness is 3. You have 3 species: dandelions, daisies, and columbine. And what's also important to notice is the total number of plants in each one of these plots is 1,000.
Welcome to ESM
So
101 Lab.
this.
My name is Alex
Same
Staunch, and I'll be
species,
the
same
graduate
number of
teaching
species,
assistant
same number of
for this
individuals.
course. So
Now,
basically, I'll be
evenness
administering
is basically the
narrating
ratio
lectures
of one
that could be a
species
good
to another,
or bad thing,
or the relative
as well
abundance
as
of the different
I will
species,
be monitoring
making up the richness
discussions,
of an area.
grading any material,
So in plot number 1,
and
if we
also
look at
fielding
that,
any questions.
you
or
have these
concerns
individuals
you
that are all
have
basically
for the lab section
present in
specifically.
the same amount.
So you can find my
Their
email
numbers are basically
on the
the same.
D2L
They all total to 1,000.
module.
While plot number 2,
it's right in the
you have
syllabus, as
columbine
well
being by
as
far
you can message me
the
directly
dominant
on
species
D2L.
in this area.
So I look forward to working with you throughout the
So
quarter.
this can be
and
seen as
hopefully
columbine
you get a
could be
lot
invasive
out
in
of
this
this
area,
lab as well as the ESM
kind of
lecture.
taking out dandelion and daisy and competing with it, or there could be inadequate sources for dandelions and daisies compared to a columbine. But in this case, I just want you to think, is the diversity the same in these areas, even though the number of species and the total number of individuals are the same? And of course, most likely you're thinking no. Plot number 1 is much more diverse than plot number 2. But one way other than just eyeballing it is to actually calculate the diversity. And there's numerous ways to do this. One being the Shannon's Diversity Index. Now this isn't 100% necessary for you to do. I just want this to be available for you so you could possibly use it when you visit your field sites, when you're trying to come to conclusions during the class-long study that you're about to complete. But also it provides you with a better understanding on how diversity is viewed, as well as I just always like to present a quantitative example of something I'm trying to explain. So this right here is actually the equation for Shannon's Diversity Index. And H being just the value that stands for Shannon. So if you see H equals 2.61, that's the diversity of that area. And then S, which is this kind of mutilated symbol up here, stands for the total number of species or richness. And then PI is the proportion of a specific species compared to its overall abundance. So here I'm using an example from the table that was on the previous slide. So plot number 1. So we're looking at dandelions, daisies, and columbines still have the same number. And whenever you see N, it stands for the number. It's that simple. So dandelions 330, daisies 320, columbines 350. And then P, or PI, which is what this is referring to, is their proportion. So dandelions at 330 individuals out of 1,000 make up a proportion of 0.33. Same with daisies and same with columbines. And then this symbol right here stands for the natural log of that proportion. So you can basically calculate this by utilizing a graphing calculator. Even some most basic calculators have this function. And there's also sites online that you can just put it into a natural log calculator. So I basically acquire the natural log of this value, and this is what I came out with. And the next step here, this function is to multiply that value, the natural log of the proportion, times the proportion. So negative 1.1 times 0.33 yields 0.366. So once you've done that for each and every one of your species, this little symbol right here means that you add them all together. So you would take 0.366 times and 0.356 and 0.368, which are all negatives, and add them up, and you get negative 1.089, which then in the Shannon's diversity index you multiply by a negative value, and then you would be yielded with a positive 1.089 for your diversity of that area. So I would like you to attempt this calculation with plot number 2. You can make a table just like this, and you can use those values, and the answer, basically what's yielded, is 0.435. So I want you to be able to compare it, and you'd be able to find out if you did it right or not. And also take this a step further and play with the numbers. Add more species, make them just as even, if not less even, like add another species and make the proportions all 0.25. See what that does. See if that increases the diversity or if it stays the same, if they're basically even. So yeah, I just want you to be able to overall realize what diversity is at a qualitative and quantitative viewpoint. And please feel free to contact me with any questions regarding this lecture if you need clarification, or if you even thought that I talked too fast or too loud. Just let me know. And yes, leaving you with the fact that more individuals compared to less individuals does not mean a better ecosystem. So get to look at diversity around you, and hopefully this allows you to view your field site when you pick it out in a different way. Thank you.